Artificial cell engineering using vesicles is an exciting field at the intersection of synthetic biology, materials science, and biomedical engineering. In essence, researchers design and create vesicles, tiny spherical structures formed by lipid or polymer bilayers that mimic the compartmentalized structure of natural cells. These vesicles can be thought of as artificial cells, providing a controlled microenvironment where biochemical reactions take place, much like they do in living organisms. One key advantage of using vesicles is their ability to encapsulate various biomolecules such as enzymes, nucleic acids, or drugs. By adjusting the composition and properties of the vesicle membranes, scientists can engineer systems that respond to environmental triggers, such as changes in pH, temperature, or the presence of specific molecules. For example, vesicles have been designed to release their cargo only when they encounter a particular stimulus, making them ideal for targeted drug delivery. How does this improve therapeutic outcomes? It minimizes side effects and ensures that the active agents reach the exact location where they are needed. Another fascinating aspect is the use of microfluidic techniques to produce vesicles with precise control over size and composition. Microfluidic devices allow for the generation of highly uniform vesicles, sometimes referred to as liposomes or polymosomes, that are essential for reproducible experiments and applications. In research, these well-defined vesicles are used to study fundamental processes such as membrane fusion, transport, and cellular signaling in a simplified, controllable setting. Can you imagine the potential for studying complex biological processes in such a streamlined system? The answer is that it not only accelerates basic research, but also guides the development of innovative biotechnologies. Artificial cells engineered from vesicles are also finding applications in the development of biosensors and artificial organelles. By incorporating specific receptors or enzymes into the vesicle membranes, scientists can create sensors that detect environmental toxins, pathogens, or metabolic changes with high sensitivity. Moreover, researchers are experimenting with assembling multiple vesicles into larger networks that mimic tissue-like structures, paving the way for breakthroughs in regenerative medicine and tissue engineering. How could this technology reshape medicine? By providing customizable, living-like systems that can adapt and respond to the body's needs, leading to more effective treatments and diagnostic tools. Furthermore, the field is exploring the creation of self-replicating artificial cells. These systems aim to emulate one of the most fundamental properties of life, self-reproduction, using vesicle-based constructs. Though still in the early stages, such research holds the promise of understanding the origins of life and developing synthetic systems that can perform complex functions autonomously. Isn't it remarkable to think that we are on the brink of creating lifelike systems from scratch? The answer lies in the clever engineering of vesicles, which serve as the building blocks for these artificial entities. In summary, artificial cell engineering using vesicles represents a transformative approach to replicating and harnessing the functions of living cells. Through the precise design of vesicle membranes and the use of advanced microfluidic techniques, scientists are developing systems that can encapsulate, protect, and deliver biological components in a controlled manner. From targeted drug delivery and biosensing to fundamental studies of cellular processes and even the creation of self-replicating systems, vesicle-based artificial cells are opening new avenues in both research and practical applications. This innovative approach not only deepens our understanding of life at the molecular level, but also promises to revolutionize future biomedical and biotechnological solutions. Artificial cells mimic biological functions by encapsulating biomolecules within lipid or polymer vesicles, enabling controlled biochemical reactions. These vesicles serve as minimal models of living cells, allowing researchers to study membrane dynamics, molecular transport, and synthetic life processes. Their design integrates membrane-bound compartments with functional biomolecules such as enzymes, ribosomes, or DNA, enabling replication, metabolism, and signal transduction. Researchers have engineered lipid vesicles that encapsulate synthetic transcription translation systems, producing functional proteins without living cells. This is similar to building a self-contained miniature factory where raw materials are converted into useful products within defined boundaries. Lipid vesicles, or liposomes, replicate the properties of biological membranes by forming bilayer structures that control molecular exchange. Their amphiphilic nature allows selective permeability, enabling the incorporation of membrane proteins for controlled transport and communication. Modifications such as pegylation or cholesterol incorporation enhance membrane stability and regulate interactions with external environments. Lipid vesicles with embedded ion channels have been developed to mimic neuronal signaling by allowing controlled ion exchange, simulating electrical activity. These vesicles function like gated security systems, selectively allowing passage based on predefined conditions. Polymersums, synthetic vesicles composed of amphiphilic block copolyms, offer superior mechanical stability compared to lipid-based vesicles. Their tunable properties enable precise control over vesicle permeability, degradation rates, and response to stimuli such as pH or temperature. Functionalized polymersums can encapsulate enzymes, drugs, or genetic material for targeted biomedical applications. Polymerosomes have been used to deliver CRISPR-Cas9 components into cells, facilitating gene editing with improved efficiency and reduced immune response. These vesicles act like durable, programmable cargo carriers that release their payload under specific environmental triggers. Artificial cells enable the reconstitution of metabolic and signaling pathways by encapsulating enzymes and cofactors within vesicles. By recreating biochemical cascades, researchers study self-sustaining reactions that resemble natural cellular metabolism. Integrating ATP synthesis mechanisms within vesicles enables energy-dependent reactions crucial for cellular mimicry. Scientists have encapsulated glycolytic enzymes within vesicles to create artificial cells capable of breaking down glucose and generating ATP. This system is like a miniature power plant where energy is extracted from fuel sources to drive essential processes. Artificial cells interact with biological cells through vesicle-mimetic communication, enabling signal exchange and coordinated responses. Functionalized vesicles can release signaling molecules, trigger immune responses, or influence cellular behavior through membrane fusion. Synthetic biology approaches integrate genetic circuits into artificial cells, allowing them to respond dynamically to environmental cues. Engineered vesicles containing quorum-sensing molecules have been used to regulate bacterial biofilm formation by altering microbial communication networks. This process is similar to sending coded messages between different teams in a large organization to coordinate collective behavior. By encapsulating nucleotides, polymerases, and ribosomes, artificial cells can support genetic replication and transcription translation processes. Advances in cell-free expression systems enable controlled protein synthesis, providing insights into early life evolution and synthetic biology. The integration of genetic self-replication mechanisms represents a key step toward artificial life capable of evolution and adaptation. Artificial cells have been designed to transcribe and translate simple genetic circuits, enabling the autonomous production of functional proteins. This is akin to a self-copying machine that not only duplicates instructions, but also constructs functional components based on them. Vesicle-based drug carriers enhance therapeutic delivery by protecting bioactive molecules from degradation and ensuring controlled release at target sites. Functionalization with ligands or antibodies enables receptor-mediated uptake, improving selectivity and reducing side effects. Responsive vesicles can release drugs upon external stimuli, such as light, heat, or enzymatic activity. Artificial vesicles encapsulating anti-cancer drugs have been engineered to release their payload upon exposure to tumor-specific enzymes. This system works like a smart delivery drone that only releases its package upon recognizing the correct recipient. One of the fundamental challenges in artificial cell engineering is enabling controlled vesicle division to mimic cellular reproduction. Researchers are exploring lipid dynamics, osmotic pressure, and self-assembling peptide networks to drive vesicle splitting. Achieving repeatable growth and division cycles brings artificial cells closer to mimicking biological replication. Experiments have shown that lipid vesicles can be induced to divide by controlling osmotic swelling and membrane tension, simulating primitive cell division mechanisms. This is like blowing a soap bubble that naturally splits into two when external forces reach a critical threshold. Despite advances, challenges remain in achieving self-sufficient artificial cells with long-term viability. Key research areas include energy generation, dynamic membrane remodeling, and integration of artificial intelligence for adaptive responses. The next generation of artificial cells may incorporate synthetic organelles or biohybrid components, blurring the line between natural and engineered life. Recent work has demonstrated vesicles capable of self-repair, a crucial step toward developing autonomous synthetic life forms. This is similar to designing self-healing materials that repair cracks automatically, extending their functionality and lifespan.